All right. So thank you, Lila. Hi, everyone. I'm Brandon, the education chair, as Lila just mentioned. Um, First of all, this is being recorded. If you do not want to be recorded, go ahead and turn your camera off. Um, but yeah, welcome everyone. So tonight we're really happy to have Bruce Filtcroft giving us a presentation on New Jersey mycoforestry and probably a touch of agroforestry stuff as well. For anyone that has regular attendees of our Taxonomy Tuesday, they may have seen Bruce pop in a couple times and kind of give us a taste of some of the concepts that we're going to be discussing here tonight. Um, and this presentation really kind of opens the eyes to some of those concepts that we were briefly introduced to there. So, a bit about Bruce. Bruce Flitcroft, he's been a member of NJMA since 2022. He's the president and managing member of Mendham Mushrooms LLC, which is an organic agroforestry business growing wildcrafted, functional, and culinary mushrooms. Um, Bruce spent 32 years in the information technology industry, founding and leading companies. He retired to pursue his passion in organic agriculture and mycology, leveraging his background in biochemistry from his undergraduate days at NYU. In 2021, Bruce launched Mendham Mushrooms to further explore agroforestry techniques that he and his team have been researching and testing successfully. Today, Bruce leads several research projects in the areas of microbiome interactions of hardwood trees and the rhizosphere and endophytic fungi and bacteria in collaboration with Rutgers University and New Jersey's Economic Development Authority. Mendham Mushrooms is implementing scalable, wildcrafted growing techniques for various species of saprophytic and ectomycorrhizal fungi. These techniques are designed to organically produce and market fresh and dried mushrooms higher in metabolite diversity and concentration, similar to wild foraged mushrooms than current industrial growing methods might uh, imply. So, with that, Bruce, would you like to take it away?
Great. Thank you. Thank you very much for the introduction. That's a long introduction. It was a mouthful. Thank you. All right.
Yeah.
So, um, first of all, everybody's getting more and more in tune with more people we know that are getting interested in mushrooms and eating them. In the last 15 years, there's been a ton of research pouring out about the health benefits of eating different types of fungi. But a lot of people don't understand why and how those health benefits happen and how different mushrooms produce different health benefits and different ways you grow them produce different health benefits. So one of the things I started doing when I started growing mushrooms 7 years ago, I was growing them the way everybody else did. We grew them with sterile environments. I grew them in my basement and in my barn, and we used sterilized substrates, and I got really good at growing mushrooms. Then I started to really get into the biology and the chemistry of mushrooms and realized that there was something that wasn't quite right. Wild crafting is growing mushrooms the same way that nature grows them, not in this indoor, industrial, sterile environment where 99% of the mushrooms that people eat today are grown. Now, if you're a forager, which I hope everybody on this call is, the mushrooms you pick out in the woods that you eat yourself, they taste different. They taste a lot more interesting. They have more flavors. They're more pungent. They taste different than the mushrooms we get in the store, which all seem to taste kind of the same if you haven't noticed. So I'm going to start with telling you a little story about something that happened in Japan in 2000. A lot of mushroom growers figured out how to grow Graphola frondosa indoors on sterilized oak sawdust blocks in Japan. A lot of growers do it here in the United States today. Now, Japan had a pretty big market. People had been eating and hunting for Graphola frondosa in northern Japan for centuries. So when it became available in the grocery stores year-round, consumers were really excited about it. And they started buying it, and it would sell out quickly. But a lot of the old people started to complain that it didn't taste like Graphola frondosa. It didn't taste like real maitake. And this caused a handful of research students in different universities across Japan to start to test it and see why it was different. It turned out we're going to go through some of the reasons why it is in fact very different, and I'll even touch on a few of those studies on Grifola frondosa in a little while. But it turned out that nature doesn't grow mushrooms the way we do in factories, and the mushroom comes out very different. The USDA currently has, I think they just closed open questions, but they're trying to define organic mushrooms. What's an organic mushroom? Because today, the way the mushrooms that we from the grocery store are grown, they're GMOs. They tend to be grown in sterile environments with non-organic substrates. They're usually treated with pesticides or other chemicals. They have additives in the substrate to try to encourage different types of metabolites. It's a science. It's really not really that natural. And we're discovering that there are some great benefits to those mushrooms, like all the beta glucans and chitins and some of the metabolites, but they're not the same as the ones we pick in the woods, even if it's the exact same species and even if it's the same strain. If you buy it in a grocery store and you pick it out in the woods, it's a very different mushroom. And that's what this deck is going to try to walk us through to understand and why that's important. So let's see, next slide, come on. There we go. So the FDA, the FDA measures what's called primary metabolites. We all know them as proteins, carbohydrates, fats, vitamins, minerals, alcohols, and lactic acid. These are what in 1890 we figured out were the compounds that you use to make cells and power cells. So the science of 1890 was the biology of life was based on primary metabolites. That if you had primary metabolites in these categories, you could make a living thing and power it, and that's what we all needed in our diet. So you'll notice the labels, all the nutrition labels and facts and everything, basically only talk about and measure primary metabolites. So the mushroom industry only had to worry about primary metabolites in the stuff they grew. But mushrooms, fungus in general, working with bacteria are the chemical or the organic chemical factories of the planet. Most of the complex organic compounds in the planet are synthesized between bacteria and fungus. So what we get is they manufacture all these secondary metabolites, and there are tons of them. For instance, we've already discovered about 15,000 terpenes and 60,000 terpenoids and about 6,500 flavonoids. And if you look at polysaccharides and glycoproteins, there's a limitless number of variations that can be created in nature, just because there's such large compounds with so many folds and so many things attached to them. That's where all the interest and excitement about eating mushrooms is coming from, is all of these secondary metabolites. Because secondary metabolites don't make cells and they don't power cells. They regulate them. This is how all the chemical reactions in your cells are regulated, is with compounds like these. And our FDA doesn't measure them, and we really never focused on them in Western, diet or science. It's really only been in the last 20 some odd years that we've really been studying secondary metabolites. And we're finding out that these are the compounds that regulate our endocrine systems and our neural systems and our digestive systems and all of the different functions in our cells are regulated by secondary metabolites. And there are hundreds of thousands of them. And most of them are manufactured on this planet by fungus. So it turns out the key to a lot of health issues may lie in all these secondary metabolites that are in fungus that we don't measure in our dietary systems. The FDA doesn't have any measurements for them or care about them. There's no regulations around them. So if you produce a mushroom that doesn't have many of them in it, it's the same mushroom as one that has tons of them in it. So that's the initial concept that we're going to get into the hows and whys in this presentation. So any questions pop up so far, Lila? No? So if you have any
Yeah.
questions, just put them up in the chat, because I don't want to start losing people as we get through some of this. But the first concept is secondary metabolites are where most of the interesting chemistry inside fungus happen. Now, there's another concept. When I was in middle school taking biology for the first time, I was introduced to this concept of symbiosis. It was a really cool, simple concept that I could understand. It was really romantic. It had the idea that two organisms could help each other through life. One gives the other organism something it wants, and the other organism receives something and gives it to the other symbiote. And that this makes life better. Kind of sounds like a marriage. It's a very human, very human, romantic notion that we can grab and grasp really easily as kids, that we really use to focus how we think about symbiosis, even as adults and even as we go through our college studies. Turns out we're discovering that that's not how we should think about symbiosis. We should probably be introducing it as tripartite biosis, which is at least 3 organisms participating. Because most of the symbiotic relationships we're discovering as biology and ecology are advancing in the last 2 decades is that there are usually 20 or 30 or 50 or 100 different species interacting at once to create an environment that becomes balanced. It turns out 2 species alone almost never create a balance in any ecosystem. It's working together, and it's because of the underlying chemistry. Archaea and bacteria made terrestrial life possible on this planet. The reason is they're able to break very hard chemical bonds, like N2. They can take nitrogen, tricovalent bonds, and break them. No other life form on the planet can. Same thing with potassium and phosphorus and other things that find very stable forms. We have to be able to release that for life to make organic compounds. And it turns out archaea and bacteria can break those compounds, and everything else evolved using it. For instance, you can't make proteins without nitrogen. So we don't have a way ourselves of getting nitrogen out of the atmosphere, but bacteria do, and archaea do. So what we see is that the symbiosis starts with an archaea and bacteria, and then what happens is the fungi use it. They create new compounds, and then they trade it with plants, and plants add to the compounds or modify them again. So we start seeing that there's not one species involved in any symbiotic relationship. It's usually 3, 4, 5, 8, 10. And sometimes it's multiple types of bacteria or fungus will do different steps of processing a chemical until they get to an end state. The production of a chemical, as we know in a lab, may take 20 or 30 steps. In nature, it also takes 20 or 30 steps. And it might take 20 different species to participate to make one of those really complicated polysaccharides. So that aha moment about 20 years ago in science really started to make us realize that these biomes are really where the chemistry of nature happens. When we got done with the Human Biome Project, we couldn't explain why the premises of the Human Biome Project turned out to not be true. Does everybody remember? I don't know how old most of you are, but when they launched the Human Biome Project, the reason was, as soon as we can study the DNA of every human, we'll see the differences in genetic deviation from human to human, which will explain why one person gets sick and another person doesn't. Why one person contracts this disease and another one doesn't, or one can be cured of it and another one can't. And that was the concept of all the funding. But when they got done with the project, they sequenced the first human, and they went out and did another 20,000 humans around the planet. They found that genetic deviation didn't explain any of the diseases and illnesses that we really had, except for a very small fraction of them. It turns out we were much more similar genetically across every human on the planet than we ever imagined. the genetic deviations were such a small fraction of our genetic makeup. So, and people with the same genetics, were one would get sick and another one wouldn't. And we came to realize it was about genetic expression, not the genome itself. And we started to understand that genetic expression was how we interacted with our environment. Every test that was done for the next couple of years was inconclusive because for human for human medicine, for genetic expression, because we came to the realization that it was our biomes that were actually interacting. It wasn't our DNA. It was the DNA of the fungus and the bacteria and the phages and everything else in our bodies that was participating. It was a far more complicated system than just, our genes helping us react, chemically to an environmental pressure. So here's, so actually, I don't know why this slide, I should have taken this one out, but flavonoids are a good example. There are over 6,000 different flavonoids, and we know them primarily as they help create flavors in our food, but they're also antioxidants. They're all antioxidants. So there's over 6,000 identified so far and counting. And most of these flavonoids are either developed by fungus or by plants from compounds that the fungus gave them or traded them. So it's amazing how many of these things we're realizing that flavors in nature have a lot to do with how plants and fungus interact. And flavonoids are not just for flavor. They're actually help, control different chemical reaction platforms. So let's get to our biome. There's three microbiomes in the human body, right? The first one is our gut biome. That's the one everybody seems to understand and talk about. In a healthy human who lives outside a lot and interacts with nature and goes on a lot of forages and eats a lot of wild mushrooms, they probably have about 1,500 different species of bacteria in their gut and about 700 species of fungus. In comparison, if you live in a big city and you live in a house a lot and you use a lot of disinfectants in your house, you probably have about a third that many in your biome, in your gut biome. Probably have fewer on your skin. A healthy person should have about 80 different species of fungus living on their feet. But most of us have far less than that. It turns out our feet were for millions of years our first contact with nature, and the first line of defense was a lot of fungus that would protect us from everything we'd come in contact with. So anyway, that's part of our skin biome, which is the second biome. The third one is the respiratory oral biome, and then part of the skin biome, there's an extension for women. There's the uterine vaginal biome, which is an extension of their skin biome, which is different than on men. So think of it as three major biome systems with thousands and thousands of participants, all contributing to chemical reactions, making secondary metabolites and other compounds that our systems use to either regulate our bodily functions and what our cells do, and also act as our first line of defense in how we interact with the environment around us. So the next slide looks at trees. It turns out trees have three microbiomes. The first one is the rhizospheric biome, which is where it collects its nutrients and protects the tree from macro attacks, and it also creates a lot of the symbiotic relationships and encourages bacteria. This biome is almost identical and shares a lot of the same species as our gut biome. If you look at the endophytic biome in the tree, which is, I'm going to jump ahead here. If you look at a cross section of a tree, this area right here, the inner bark and the cambium, basically, and the phloem, this living area of the tree, is full of fungus and bacteria. All endophytes, which are creating a lot of secondary metabolites for that tree. That's where its immune system actually comes from. The immune system actually is from those bacteria and fungus creating tannins and all sorts of things to discourage insects from eating them. They create the compounds that make the bark fire retardant. They do a lot of things. Then the third biome is the phylospheric biome, and this helps encourage growth. This is actually the bacteria and fungus that live in the tips of the plants and on the leaves that help with photosynthesis and help stimulate growth of new cells. So it turns out their three biomes are very similar to ours. Nature seems to have a system that we're discovering, that it has these complex microbiomes and systems for different functions that all higher life forms have evolved utilizing. And we really didn't put a lot of time and energy into researching this for the last couple hundred years. Science really only started appreciating it probably 30 years ago, in the 90s. And it's the advent of all the genetic technologies that are coming out that are making us be able to see and understand how this works and why it's so important. So, all right, here's a picture. This is one of the rooms at the lab here at Mendham Mushrooms. This is an old carriage house I rebuilt on my property and turned into a laboratory. That's me in the back. Anyway, so let's take a look at something that you've probably never seen. If you were to look at the hyphae of the fungus that you pick in the woods, these are samples that were grown here at Mendham Mushrooms. We took them down to the labs at Rutgers. We stained them and we blew them up. And these are endohyphal bacteria, a term you probably never heard of. It has a lot to do with fungus. It turns out fungus invite bacteria into their cells, lots of them. There's a lot of different species, and because these bacteria are producing the raw materials that the fungus are using to make more complex organic compounds, like those secondary metabolites. So here's a highlight. You'll see all these little dots here. Those are bacteria inside the hyphae of a fungus. Now here, the fungus actually allows the bacteria to travel in and out of the cells. These are bacteria emerging, coming out of a hyphae. So try to imagine bacteria being invited in and out of the cells of fungus. Why do they do this? There's a couple of reasons. A few of them are theoretical, but the first one goes back to one of the things I talked about earlier, which is in fixing compounds, right? So let's take nitrogen. Everybody's heard of nitrogen fixing. So fungus can't fix nitrogen. Neither can animals or plants. We need bacteria or archaea to fix nitrogen. So they take it out of the atmosphere and they break it down into individual nitrogen molecules and they attach it to something else, whether it's they make a nitrate or an ammonia or some other organic nitrogen compound that's soluble and usable to create life, right? So that's one reason. So fungus can't create these, release these really hard bonded compounds like phosphorus, potassium, and nitrogen. So they invite bacteria into their cells to do it for them. And you'll see some pictures of that in a little while. So when a bacteria is invited into a mushroom cell to release a compound, let's say, I'm going to stick with the nitrogen example. The problem is it releases lots of oxidative compounds, particularly like oxygen, right? Oxygen prevent fixation of nitrogen or negatively charged ions. So it turns out the mushroom is producing terpenoids and polysaccharides, and the theory goes they're sweeping up the excess negative ions and the byproducts of the fixation. So if a mushroom wants to make more hyphae and more cells, it needs to create proteins and chitin and beta glucans and things, but it needs a lot of nitrogen and phosphorus and other compounds that it gets from the bacteria. But the bacteria needs to do it efficiently because we all know how fast mushrooms want to grow in the wild. So they create these other compounds to keep sweeping up all the waste from these chemical reactions from the bacteria so that the bacteria can just keep producing more and more and more nitrogen and phosphorus and all these other things that they need. So the bacteria is giving them the building blocks to create these complex compounds and like glycoproteins and proteins and everything else. And it's also the mushroom is generating these really complex secondary metabolites to regulate the chemical reactions, to be able to prevent cells from growing out of control, to prevent oxidation of cell damage, to protect the bacteria and to sweep up these excess chemicals. Now, doesn't that sound like a lot of the problems we have in our own medical, problems these days? We're all looking for antioxidants, right? That's been the new craze the last few years. The reason is we realize we have too many oxidative compounds in our diets, and too much chlorine and fluoride in our water and everything else. So we need antioxidants to sweep it up and clean it up in our systems. Turns out mushrooms do this naturally with bacteria. They get the bacteria to produce the building blocks for them, and then they create the compounds to keep the process as efficient and fast moving as they can. So the theory is that the presence of different species of microorganisms trigger the fungal genes to trigger production of a wide range of secondary metabolites. This is back to gene expression. The mushroom won't create many of these secondary metabolites unless it senses the presence of the bacteria species that it is going to try to get this chemical process going with. Does that make sense to everybody? That took me a while to really grasp and get my head around for a while. But if I were to grow a mushroom in my basement with no bacteria in the substrate, it's like you wouldn't build a building unless you knew you had the labor and all the materials. You wouldn't contract it. You wouldn't spend the money. These are very expensive compounds to make. They're big and complicated. So the mushroom's not going to make them unless it knows the bacteria that's going to give it all the raw materials is there. So what we see is that there's a gene expression in the mushroom that when it comes in contact with different other species of fungus or bacteria or archaea or protozoa, it'll now start creating all these complex organic compounds. So that's, here we go. I think I just explained half this, or all this slide. Yeah, so yeah, antioxidants like terpenoids and polysaccharides are sweepers of oxygen. And I explained the gene expression theory as well. Now, we don't have proof of the gene expression in fungus. This is a new field, a new area that really is just beginning the study of. Because as soon as we figure this out, we're going to be able to identify, what bacteria and archaea and fungus create these different compounds that trees, plants, animals, humans use to regulate the different systems in our cells. This is just the beginning of this science. So, which brings me to one of my primary collaborators, Dr. James White here in New Jersey, is head of the plant biology department at Rutgers. He's got a lab down there, and his thing is mycology. He's really a mycologist at heart. He spent a lot of years doing research on vascular plants and arbuscular mycorrhizals. It had a lot to do with the USDA changing a lot of the strategies of how we want to grow crops, field crops in the future. We want soil health to improve to have arbuscular mycorrhizals. Well, Jim's been switching his focus and his graduate students' focus over toward different types of saprophytic fungi, not just arbuscular mycorrhizals, because they're really starting to get into exactly the concepts that I had talked about in watching the interactions between bacteria and fungus and the chemistry that's going on symbiotically and how there's so many different species participating. There's not one species of bacteria that gets invited into a cell. If you want to make a glycoprotein, it is thousands of molecules in size usually, or a polysaccharide. There's a lot of different materials needed to make it. So there's different bacteria that are working with fungus, and sometimes multiple types of fungus working together to create these compounds. And again, why is that important? Because that's where the immune system of a tree comes from. That's where the growth incentive of cells of plants comes from. That's where, there's so many things that life is dependent on that we didn't realize that fungus are really the organic chemical factories of the planet for life, particularly terrestrial life. So here's some of the things. Here at Mendon Mushrooms, we've been providing some samples to Dr. White's lab of wildcrafted or wild mushrooms. So here's an example of a reishi mushroom that, interestingly enough, we don't just see here. This is an example of the lateral emergence, again, on the left, of bacteria in and out of the hyphae cells. But on the right, on this next slide, here you go. What's interesting is we're seeing certain bacteria being moved into the basidiospores. It turns out that these bacteria might be helping these spores in ways that we'd never imagined, because Dr. White's lab is realizing that they're finding bacteria inside the spores, and that once the spore is fully developed, you can't see it anymore because the outside of a spore is really hard, and the microscope can't see the difference in the bacteria. But when it's very young in the basidiospore early stage, the cell walls aren't developed really hard with all that chitin, so you can still see the bacteria entering into these spores before they're released. Now, this one's a chlorophyllum, but that's a concept that was kind of newly discovered by Jim and his team, and it says a lot, because if you think about it, in humans, the vaginal uterine extension of the skin biome in females passes tons of fungus and bacteria onto the child to provide its initial defense system, its initial immune system. There's a possibility that this is doing the same thing to get that rapid growth of cells and hyphae on an emerging fungus. It might need this bacteria to help be the engine to drive the access to nitrogen or phosphorus or whatever it needs. So again, this is new. This is kind of new discoveries, and we're not exactly sure why it's happening, but there's some really cool theories around it that need the research to prove them or disprove them. So here's a nodule of hyphae loaded with bacteria. Now, this one in particular, we're trying to figure out all the different species of bacteria in this nodule. We were able to identify 5. The problem is we need to start doing metagenomics testing to discover them all, which is what we're doing. We're applying for 2 grants this year to do 2 large-scale 2-year research projects in order to identify metagenomically all the different species of bacteria and fungus that are working together in a microbiome, because we would like to know all of the different ones that are in here. So I'm going to circle back to something I introduced early in the speech, which was those guys in Japan, those old people in Japan that were complaining, these don't taste like maitake, right? So there were a handful of universities that tried to figure out why the maitake grown for the grocery store was different tasting than the ones they picked out in the wild. So the first thing they did was clued in on taste. The science of taste has been around for a while. It was pioneered back in the 60s. And we know a handful of things. One, there's a glycoprotein called a group of proteins called 5 nucleotides, like 5-CMP and 5-GMP. These trigger our taste buds to emphasize the reaction, and it makes taste amplify. So having these glycoproteins in our food source amplifies flavors, right? So they studied that to see what the results of these different glycoproteins were in store-bought or grown in sterile environments versus grown out in the wild and on logs and in the soil. The second thing they looked at was the total amount of proteins in chitin. And the next thing that affects flavor a lot is free amino acids. right? Different free amino acids left over from the production of proteins give us different flavors in our mouth, right? And then there's a handful of glycoproteins that are only found in they give us that umami flavor. They're really only found in the cell walls of a lot of animals, so meats. So we get that meat flavor from mushrooms. It's because there's certain glycoproteins that that mushrooms produce that are the same ones that are used in animal proteins. So what did they find out? They found out that the total amount of glycoproteins in the 5 nucleotide range were much higher in the wild mushrooms that they grew on logs behind the university out in the woods versus the ones that they grew in a sterile environment in the lab the same way you would grow at one of the big mushroom producers. The second thing they realized was that there was the exact same number of proteins and chitin and beta glucans in both samples. And this was across 2 different studies at 2 different universities. They found the exact same amount of proteins in the same proteins. So back to that primary metabolite concept. The mushrooms made the same proteins because that's what they do. But what they found was that there were a whole different mixture of free amino acids. So like the wildcrafted ones were high in L-alanine, which is a sweet flavor, right? And glutamic acid, which is a umami flavor. That's that, meat flavor. But they were lower in tyrosine and aspartic acid and others, which are the flavors of bitter, etc. So they inverted. There were more bitter flavors and no flavor at all amino acids in the sterile substrate grown mushrooms, but they were higher in umami and sweetness and stuff in the wild grown mushrooms. What it told them was that the mushrooms were using different processes to construct proteins based on what was available in their environment. And it was much more efficient to have the bacteria help them. And that process released leftover alanine and glutamic acid. Where they didn't have the bacteria to help in the production, they ended up having to use a different protein production system that left different free amino acids, creating a different flavor in the mushroom. So the old people were right. And their memory of flavors were true, that the ones grown in sterile substrates tasted different for these reasons, even though the primary metabolite values were the same. So, any questions on that? All right. So, I just covered some of this. So this is a couple of the different flavors that we had on that previous slide. And again, you'll see the top 2, there were higher concentrations in the wild ones. The bottom 3, there were higher concentrations in the sterile substrate grown mushrooms. So what we're doing at Mendham Mushrooms is trying to figure out a way to grow mushrooms the way nature grows them at scale, and create economically viable ways to do it. Because we went indoors with sterile substrates for a lot of reasons. It wasn't economically viable. You couldn't have hundreds of people out scouring the woods trying to find or forage for mushrooms and then sell them for a couple of, probably 4 main reasons. Let's see if it's on another slide. Yeah, it's probably on the next slide. Come on, there we go. There we go. So today, most of the mushrooms we all eat are commercial sterile production. You can create mushrooms year-round that way. It's indoors. You can control the light, you can control the moisture, you can control the temperature. They don't have any bacteria or archaea or phages or protozoas or anything in their materials, which gives them a long shelf life. And that's a topic we're going to talk about in a few minutes. And it made their yields predictable. They no longer had, infestations of bugs, or they weren't eaten by slugs, or the deer didn't come and eat them overnight, right? But the results were what we didn't realize, because we weren't measuring secondary metabolite. We were creating a less nutritious product. All those really cool compounds that help us regulate our immune system and our endocrine system and our hormones were actually not being produced, or as many of them anyway, in the commercially sterile grown production. There's a much wider range of those compounds and much higher concentrations in wild ones. And they taste different. They don't have all the flavonoids, and they don't have the same free amino acids in them. And, they taste different. They taste less. Have you ever noticed that the mushrooms in the grocery store all kind of taste the same? Most people don't realize that till they've been out foraging and eating mushrooms in the wild. So if you eat a portobello or a crimini or a button mushroom, they're all from the same species, and they all taste the same. But you can also eat the maitake and the shiitakes and everything, and the enokis, and they kind of taste the same too as the button mushrooms. It's because of the difference in the compounds being generated from this lack of symbiosis of all the different species that they're supposed to be working with. So by growing them in the wild, or wild crafting them anyway, we get a fuller complement of all those secondary metabolites. They have more taste, and they get a premium price for that. People are worried about their nutrition. It's one of the big drivers, and they know that even the store-bought ones are pretty nutritious for them. But the wild crafted ones are loaded with things that we never realized could help with the regulation of so many bodily systems in our cells, and help protect us from so many different pathogens that the research is starting to come out more and more, and we're wrapping our head around it as a consumer group. But here are the problems. Mushrooms, as we all know, are seasonal. They pop up for 2 weeks. That species will pop up for 2 or 3 weeks out of the year. That's it. And the rest of the year, you don't get any. The short shelf life. We're going to talk about this for a second, because all those bacteria that they invited in and that they're working with, and they're in their cells, what happens in nature when a host organism dies? All of its symbiotes turn on it. They start to eat it. That's what happens with mushrooms. Wild mushrooms only have a shelf life of 5 to 10 days. Whereas the ones you buy in the grocery store that are grown sterile and treated with pesticides and everything else, those last for a month between the time they're picked to when you can eat them, or more. So they have a much longer shelf life. And economically, that's been the biggest driver. So going to wild crafting creates this whole downstream problem of the economics and how people buy and eat their food, back to organic, buying organic food and buying local. It turns out wild mushrooms, or foraged mushrooms, or wild crafted mushrooms have very short shelf lives, because those bacteria are going to turn, and it's not just the enzymatic breakdown of the fungus, it's also bacterial breakdown of the fungus that accelerate its going, we call it going bad, right? The third one is pests. When you're outdoors, you're dealing with the rest of nature. When you're in a locked warehouse, you don't have slugs and fungus gnats. Well, you might get fungus gnats, but you can kill them. You don't get flies. You don't get, squirrels and deer and stuff. All right, so I'm going to go on a little aside. Back to the immune system of wild mushrooms. Who here knows that squirrels collect mushrooms in the fall? They carry them to the tops of trees and dry them out on the branches in the sun. Anybody know that? Turns out they store them with their nuts inside trees and other places. But they don't eat them all winter. They eat the nuts. They don't eat the mushrooms. So why would they gather them, right? It turns out when they wean their babies off of mother's milk, the first thing they feed them is those mushrooms. That's the first food that wild squirrels eat after they're weaned. The theory is that it's the completing or promoting their immune system before they go out into nature. And that those mushroom caps were collected by their parents months in advance and stored for them. So here's another interesting fact. It turns out all placental mammals on the planet eat mushroom caps, eat 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 the fruiting bodies of fungus. It turns out we probably have much more of a symbiotic dependency on them than we realized for a lot of these immunology and metabolite, the secondary metabolite requirements for a lot of our systems. So that's just an aside. We'll come back to it. So let's talk about what we do at Mendham Mushrooms. We grow mushrooms outdoors on wood. For instance, yes, these are logs that we collect fresh cut logs because there's lots of them. Tree services, DPWs, everybody's cutting down trees. We want to use them. So here you'll see we have 3 farms that we've started in the last year and a half here in New Jersey, where we're collecting logs, we're inoculating them. Some of them we're burying, like for maitake, we're creating fields of buried logs. It's not as easy as you think. 1, logs are heavy. 2, inoculating them by hand is a pain. But really, you have to simulate an oak forest in an open field to make it work. You got to get the light right. You got to get the temperature and the moisture right. And you got to do it naturally. Different fungus fruit based on different cues is some of the stuff that we've been learning in the last few years. We all just go forage for them and look for them when they come out. But it turns out it's more than just moisture and temperature that trigger fruiting of fungus. A lot of fungus are actually cued into the tilt of the earth. You don't realize it, but there are cues, the wavelengths of light that come through the atmosphere as the earth tilts actually trigger fruiting of different fungus. They know what time of year it is based on the light gradient, the wavelengths gradients in the light coming and hitting the surface. Right? Never realized that, but it makes it, if you want to wild craft them, it makes it kind of tough to know how to grow them, because if you put them in the shade, you got to be able to simulate natural light. So you got to use natural light if you do it outside. Next, there's an interesting effect. There's a lot of mushrooms that grow in oak hickory forests. The great oak hickory forests in the world are like the eastern United States up into Canada and around the Great Lakes. The other one is in like northern Japan, Korea, and northern China. Another one is in Eastern Europe into like Belarus and Estonia and going down to the Alps. These are some of the best places in the world for mushroom foraging. There's a reason for it. Those are deciduous hardwood forests, some of the biggest and oldest on the planet. And that's where an awful lot of species of mushrooms love to grow, particularly saprophytes and ectomycorrhizals. So it turns out there's a lot of cues we're finding out doing this agroforestry, focusing on mushrooms, which is mushroom forestry. It's getting mushrooms and trees to grow together. We're discovering that things like light gradients make a big difference. What mushrooms will survive and do well? They don't like, I think as we all know, most mushrooms don't like direct sunlight, right? Here's an optics issue. If you have light come through a hole in the leaves in an oak canopy, those holes tend to be about 100 feet in the air. By the time the light hits the ground, what it does is it cones out and actually takes that amount of light and it diffuses it and spreads it out in a spectrum across a larger area on the ground and moves it as the sun's moving as it passes through the trees. So it's like a little cone of light that's spreading around, and there's lots of them, and they're moving around the forest floor. That has a lot to do with certain mushrooms and when and how they fruit and where they'll survive. We never realize that little nuances like that in the canopy and the tree selection make a big difference in why some mushrooms will grow in some areas. And it's not just light. I'm sure everybody on the call is pretty familiar with the idea how ectomycorrhizals trade with trees for nutrients, right? So the idea is ectomycorrhizal fungus like boletes and morels and chanterelles and stuff. They don't eat other life forms like saprophytes or parasites do. They trade. So they get all of their primary metabolites by either collecting it in the soil or trading with trees. So all their carbohydrates, all their sugars, all their starches, everything come from trees. And they create these complex compounds to trade with them so that the tree has an immune system and can stimulate growth and do a lot of other things. So we've seen in the last 100 million years, a lot of species have evolved specifically around certain types of trees, and they've grown accustomed to the growing environments of those trees when they're healthy. So there's other triggers for mushrooms other than just what tree it's under. But if you look at healthy groves of trees or healthy canopies, that's generally where you get the best flushes and the widest variety of different fungal diversity, because they've evolved for healthy tree environments, creating these canopies in these deciduous hardwood forests. Problem is, here in New Jersey, we clear cut all those forests 4 times in the last 400 years. So we don't have a lot of pristine natural canopy that we used to have. So it's hard to find areas where we can get predictable production of different types of species of fungus in the wild around here. We all know that spot where the chanterelles or the morels or certain boletes pop up every year because there's a couple of old trees there and they've created that little, kind of micro environment. But if you want to do agriculture like we're doing, there are not the forest structures to be able to do it on a large scale. So we're exploring a lot of methods where we can sort of duplicate and help the forest grow back in a way that it'll produce large production and scale of these fungus, where we can grow the species we want, and they'll be growing with their full, what's called the entourage effect. They're growing with all the different species of symbiotes so that they get the full entourage of chemical production within them, all those secondary metabolites. So here's a picture. So even logs that we grow in tents are still outdoors. We grow them outdoors. All these logs still have their whole endophyte set of bacteria and fungus in them. They were cut live, they were cut healthy and inoculated immediately, so that all those endophytes, remember that picture, the cross section of the tree, all those endophytes are still in there, and they're interacting with the fungus as it grows. So they're getting the full set of metabolites that they would for saprophytes anyway. And we can even do this on a smaller scale. Here's an example of building totems. We have a great use for tulip poplars. When a tulip poplar tips over and falls, we go and collect the log and we cut it into totems and we plant different types of oysters on it. And it will produce usually 3-4 flushes a year. Each totem is only about 6 feet high, and it'll produce 10 pounds of mushrooms a flush, and it'll last for 4 or 5 years. So there's a small scale. Think of small farmers or backyard projects where people might want to grow their own mushrooms in their backyard. This is a method that we do that has been around for a while. Farmers have used it for years. We're bringing it back and trying to figure out a way to make it more scalable. You don't need much. There's other types of outdoor farming for mushrooms that we're doing that are not that expensive. They don't need giant buildings and air conditioning systems. One of the challenges is we only get fruitings once or twice a year, but we get a lot of mushrooms all at once, which is not conducive to selling them in grocery stores or to restaurants. What it is good for is drying mushrooms and selling them. Food companies, there's now demand for dried mushrooms at scale that have all these secondary metabolites. There's a couple of companies that are starting to explore it, and that's probably where we're going to get our first big customers out of. The other problem is there's a lot of labor that goes into growing mushrooms and understanding how to simulate those natural environments of microbiomes and forest canopies and grove structures of trees, and preserving the endophytes in the wood that you're using. These methods are a type of agroforestry that's emerging here that we're trying to pioneer here in New Jersey. So here's some examples. All of these types of mushrooms, all the white rot saprophytes are pretty easy to do this with. So turkey, reishi, lion's mane, chestnut, shiitake, chicken of the woods. All of these we can do on logs at scale. But again, we suffer from from flies and fruit and fungus gnats. So we have to harvest quickly and regularly. If you miss a day or two, you can lose a crop. Then we dry them quickly. We sell a lot fresh. We got people, there's some local people that love our mushrooms. They come by and they'll call us, say, what do you have this week? Can I come by and buy some? But we're preparing to be able to create it for drying it for all those secondary metabolites. So now I'm going to introduce a new concept. This is a theory that's unproven that I think is fascinating. That is that now that we know that fungus invite bacteria and other fungus into their cells to do digestion processes and create nutrients, it turns out that they also create these cell walls that we know are hardened with chitin and all sorts of different proteins that are able to protect bacteria from the acidic acid in our stomach. There's a theory that mushrooms, like the skin of fruit, are nature's way of introducing our gut biome bacteria and fungus into us. So that it turns out eating organic fruits and vegetables and eating wild-crafted and foraged mushrooms are actually the most efficient way in nature's way of putting together and restoring our gut biome. There's some new research about to start on that theory. I think it's fascinating, and I think it makes a lot of sense off, just from the things I do know. I'm certainly hoping that turns out to be true, because it means a lot more people are going to be out foraging for mushrooms if that's the case. So this is my contact information if anybody is interested in learning more. And also, if anybody's interested in participating in the research that we're going to be kicking off this summer, we're going to be staking out certain stands of hardwood trees, oaks and hickories in particular, around New Jersey. And we're going to study them for a 2-year period. We're going to take soil samples and root samples. And in the spring, we're going to take samples of the buds off of them and the nuts in the fall. And we are going to test them. And we're going to look at all the species of bacteria and fungus that are living with those trees. And we're going to start over time creating a larger and larger sample and looking for all the trends. We're going to be looking at what species this, say, white oak, builds into its microbiomes that'll help it grow, and at what mushrooms are growing with it and why, and what other species are necessary to grow those mushrooms with those trees. This research is going to go on for a few years. We're kicking off the first one this June, on June 17th. We're going to start staking out our first 50, our first 50 sample sites. And we're going to be looking for volunteers to visit those sites on a regular basis, go mushroom hunting, basically. And when you find certain species of fungus that are growing there, collect it and bring it to our lab. And if you want, we'll let you test it. We'll teach you how. We'll let you take slide samples, and we'll let you put it up on iNaturalist, and we'll take spore samples, and we'll show you how to prepare the genetic testing samples. And then they'll go into our cryo freezer and then eventually down to the genetics labs at Rutgers and get results on what bacteria and fungus and trees are living together and creating these microbiomes that make up the ecosystem of New Jersey. So with that, I'm going to open it up to questions and comments. Anything you liked or didn't like about the presentation, anything you didn't understand, and if you have any questions. So Lila, are you at the controls?
I'll jump in and just say thanks, Bruce. That was good, man. Super interesting. I'm sure there are other questions, but I got a couple to start out with, if you wouldn't mind.
Why don't you kick us off with one, Brandon?
Yeah. So, this is a little bit in the weeds in terms of kind of biochemistry. So let me start with fixation. You were mentioning that.
Yep.
Can you explain that a little bit?
Sure.
Yeah.
So I had used I'd use nitrogen fixation as the example. Um, almost all the nitrogen on our planet wants to be N2. It wants to be in the atmosphere as N2 as a gas. And when it connects as N2, it has 3 covalent bonds, and it's so tight, no terrestrial life form, except for bacteria and archaea, can break that bond. So what happens is those species will actually ingest the air, and they'll break that bond because because otherwise it's an inert gas, right? They'll break that bond, and before those molecules can reattach, they attach it, they fix it to something else, like an OH molecule, or they'll make ammonia out of it, or they'll make a nitrate out of it, right? They'll connect that to another molecule that doesn't reassign, it doesn't combine back into its stable N2 form. They fixed it to something else. That's why it's called fixation.
Mm-hmm.
Now, we need nitrogen to make proteins. There's no terrestrial life form that lives without proteins. You can't make them without nitrogen. And if nitrogen's in the atmosphere as a stable compound, we can't make protein. We have to be able to derive nitrogen from less stable forms in organic molecules. And the bacteria and the archaea release it from the atmosphere and connect it to other things and fix it to other molecules so that it can be used as in organic chemistry to create life. So that's just the example of nitrogen. So
Yeah.
we say certain plants are nitrogen fixing. That's a misnomer. Plants don't fix nitrogen.
Mm-hmm.
What some plants do is they create environments where they invite the bacteria in that fix the nitrogen. For instance, peanuts are a legume, right? All these legumes, these seeds underground and stuff, are just houses for tons of bacteria, is all they really are. And the bacteria is releasing the nitrogen so that the plant can eat, can create new cells. So they're inviting bacteria, and bacteria is doing the nitrogen fixation, not the plant. But the plant has evolved in a way to invite the bacteria in the same way the mushrooms invite them into their hyphae. Does that help you understand fixation a little bit?
Yeah. Yep. It,
Okay,
it it kind of jives with, the from context what I thought it was, but yeah, that explains it perfectly. Thank you.
great. So here you go. Somebody just asked about waiting 2 weeks after cutting my oak logs. Absolutely wait 10 days. The best time to inoculate a log is 10 days after it was cut to about 3 weeks. That 10 to 21 days is the optimum time to inoculate fresh cut logs. So here's the reason. A lot of the white rot saprophytes that we like to grow on logs, think lion's mane, hen of the woods, shiitakes, etc., right? They don't kill live trees. It literally takes a week or so for the tree trunk to know it's dead. So the chemistry in the tree has to stop, and it takes a week or so. So you cannot get the mushroom to start to colonize the tree and a white rot saprophyte until it thinks the tree is dead, because it's a saprophyte. It's not a parasite. A parasitic fungus will attack the tree while it's still alive. A saprophytic fungus waits till it's dead, and the chemistry of the tree trunk doesn't change for 7 to 10 days to where it's really dead. So that's why you usually wait 10 days after you cut a tree before you try to inoculate it with a white rot saprophyte. Now, likewise, you don't want to wait more than 3 weeks, because now everything knows it's dead and it starts getting colonized by all different types of fungus. And now your shiitakes have to compete for real estate and sugar inside that log. Does that help answer your question, Hunter?
She
Okay,
says, yes, thank you.
great. Any other questions? Those are two good ones.
Well, on that kind of same along those same lines, I noticed in that one photo that I hadn't seen before, you had, I think it was pink oysters growing
Yeah.
on a totem.
Yep.
And then what is that? Flammulina maybe or something like that on vertically stacked, kind of like a like layered stacked up top there.
That is
Is there?
Namikos. Those are Namikos.
But there's an advantage to stacking those logs that way
Yeah,
for the Namikos.
you will see that a lot of them are growing on the bottom side of the logs. They actually
Yeah,
need more shade.
gotcha.
The
Cool.
oysters don't care. The oysters are easy to grow. Just put them on a totem in the woods and they grow. But
And
Nabicos are a little pickier. They don't like to grow in any direct sunlight. They'll like to be on the north side of logs or on the bottom side of logs. So to increase the growing area, we create these log cabin stacks. It works pretty good.
yeah, that's what I was actually gonna call them, log cabin type stack.
Yep.
Um, and then that photo you showed of the bacteria within the basidium,
Yep.
the cells of the basidium, um, under the microscope, are those observed to be kind of moving around and
Oh
wiggling?
yeah. No, they're alive.
Yeah,
They're
yeah,
moving
yeah,
and alive.
yeah. Because I think I've seen that before, and when I look at those basidium that are kind of wiggling around, it always freaked me out. I didn't know what it was, but I guess it's just bacteria. It's
Yeah,
cool.
bacteria in or on it. Yep. Doing a lot of the work, creating the raw materials for those mushrooms to create more complex organic compounds.
Very cool. Thank you.
So I have a question. Does everybody know about sugars and growing mushrooms? That's one of the things that I guess it's fairly well known, but I should kind of cover it here just in case. So we only harvest logs from November through March. There's a reason for that. There's also a reason why those three regions of the world I talked about have so many different mushrooms that grow and do well in them, and so many nutritious mushrooms, right? It's because deciduous hardwood trees have this really interesting behavior. Before they drop their leaves, they store tons of sugar inside their sapwood, right? They do that in the fall. All the photosynthesis they're doing is no longer growing new leaves or used for the energy to grow taller. They do that in the spring and early summer. Later in the year, they're stocking up their sapwood with sugars and starches. And by the way, those endophytes, the endophytic bacteria and fungus that live inside that tree are eating a portion of those sugars. But it's loaded up for the winter. The reason is in the spring, when it has to leaf out, when it buds and leaves out and has to start growing, it doesn't have photosynthesis yet. So how does it do all that work? How does it have the energy to create leaves before it captures light? What it does is it's using up this sugar. It uses this sugar and converts it and makes it the energy source that produces the buds and leaves out. So these deciduous hardwoods are really good at storing lots of sugars in their sapwood, not in the heartwood. The heartwood's usually packed with resins and all sorts of tannins and things. But the sapwood is left hollow and hollow tubes in it for storage of sugars during the winter. When we inoculate logs, the goal is to get the mycelium through the bark, through the inner phloem levels, that whole living layer of the cambium and the inner bark and everything, and into the outside bit of the sapwood, because there it comes in contact with all the endophytes that it needs, its bacterial partners. It also gets access to the sugar inside the wood. And what you'll see is when it takes over and starts to eat the wood, particularly the white rot saprophytes, they'll focus on the sapwood where all the sugars are, and they'll colonize very quickly. That goes back to the idea of when you cut down a tree, you wait a week, but then you're in a hurry to inoculate it after that 10-day period, because now everything's racing to eat the sugar in that sapwood or get access to it. So if you inoculate the tree with enough pieces of mycelium at the right time, and you can get it to come in contact with both the cambium and the sapwood, that fungus will start to colonize very quickly using the sugars that are left in the sapwood. Otherwise, it has to do much harder work. It has to exert a lot of energy to break down cellulose or lignin, which it'll eventually do after it's strong and colonized the wood. But to really colonize it and take off and gain territory, the sugars are much more efficient. The problem with that is everything is good at using sugars. Only certain, particularly white rot saprophytes, are really good at breaking down lignins. Most organisms aren't, but everything eats the sugar. So when you have a nice, healthy tree cut in the winter and it's loaded with sugar, pretty much everything can grow in it very quickly if it gets access to those sugars, which, I don't know, Hunter, if you wax the tops of your dowels after you inoculate a shiitake log, that's to keep the molds and the mildews and other types of spores out for a few weeks, giving the mycelium you put in there a head start to eat some of that sugar and start colonizing first. I don't know if that was helpful, but it's a little explanation of how we use logs from the wild to grow things instead of using, sterilized sawdust. And that's not new. That's been around for hundreds of years. They've been using those techniques in China and Japan for hundreds of years. All right.
So, Bruce, Lila here. So I guess you're saying with that, since you're harvesting the logs November to March, or your DPW and arborist partners, I guess you yourselves are not doing it. So all your inoculations, your totem building, all of that is happening then basically.
Yeah, we just wrapped up most of it.
Yeah, late November.
Actually, we start inoculating a lot of oak through the winter because we bury it. We'll particularly maitake. We have just to give you an idea, for each acre of land, we're started in forest, but we're working on doing it in converting cornfields into mushroom farms. It's a little more complicated back to those light and moisture and temperature issues, but we did our first acre down in central Jersey. We got 620 tons of oak inoculated and buried this year on our first acre test down there. That's on a 1,200 acre farm. If we get to 15 acres, we'll be the largest maitake producer in the world.
Yeah. So what? Wow, I'm trying to picture what that looks like. Is that like a log every, the spacing of those logs every 4-6 feet?
Less than that. Every 2.5 feet, you got a log, and they're 8 feet long, and they weigh about 1,000 pounds. And you got logs laid out in rows and buried. We make sure we get the right soil combinations, the right bacteria in the soil, and then we have to manage the surface temperature, because the soil in cornfields is very different than the soil in oak forests. So we've had that challenge to deal with. I already talked about how the lighting makes a big difference. So we've got to simulate oak canopies on the ground in a cornfield, and that's not been easy. That's a engineering problem for that's an optical engineering problem that we've got a couple of guys who are pretty smart in that category that they're working on it. So we're trying to convert typical pasture farmland into potential outdoor wildcrafting, mushroom farms.
Okay, and you're not trying to simultaneously grow any of these trees adjacent to this. You're literally keeping.
We are.
No, you are. You're also planting trees.
We're planting a lot. We plant a lot of trees, all oaks and hickories. We plant a lot of oak and hickory trees.
Okay.
So mycoforestry is growing trees and mushrooms together. It turns out one helps the other one grow.
Yeah, it's a challenge starting, I would imagine, from a cornfield, because they've been abused for
terribly abused.
Yeah.
So we start with tons of wood chips and getting the carbon levels back up in the soil, and we're collecting soil from healthy forests and trying to grow the right bacteria and introducing it into the fields. And it's a lot. There's a lot to it. It's not just, bury a log and grow a mushroom.
Mhm. No.
All right. Do we have any other questions?
I think I've got everybody activated so you can unmute yourselves. If somebody has a question and can't unmute, put a comment into the chat and I'll get you unmuted.
Yeah, Eric has a question in the chat. Eric, you want to ask that one?
Hi, Bruce. Let me get to my question here. Give me 1 second. Basically, I was asking, what will Mendel Mushrooms do with the data it collects from the metagenomic testing that you spoke about in the, related to the grants? And
Well, if it's funded by the NJEDA and it's done with Rutgers, it'll become public information. So the metagenomic information will be made public with the USDA and through Rutgers. And there's a couple of grad students we hope eventually will include it in the publishing of their PhDs in a few years.
great, thank you.
Can you touch again, just following up on that question by Eric? So you get that data, it becomes available if it's been funded and so on. But tell us again how you're going to use that to improve this, I guess, as a commercial venture.
Well, it starts as just fixing the forests. It starts as the puzzle pieces to start regrowing native hardwood forests back here in New Jersey, and to be able to grow mushrooms more effectively. Then the other work we're doing in other areas, trying to create simulated growing environments with the full biome structure, is how we plan on mass producing it, because it's really hard to just grow in the woods and travel around and hope you find the mushrooms as they come up. It's much easier if you can do it all in one place and be able to manage the pests and things more effectively. So the goal is to take a wild crafting, or the wild process that we don't fully understand yet for each of these mushrooms and each of these trees, learn more about it so that we can simulate it and start reproducing it back in our forests naturally. So one of the other hopes is that we will have reforestation projects come out of this, where communities or landowners can more effectively reforest their properties in a way that the healthy forest returns. Here's just a piece of information. I actually like telling the story. So last summer I was on the EPA roundtable in Washington, D.C., and one of the heads of the EPA just made the comment to me. He goes, the billions of dollars we've all spent since 1970 in trying to grow forests. We created all these plant a tree, and we Earth Day, and we re-promoted Arbor Day and told everybody just plant a tree and the earth will cure itself. Because the dirty secret is all the billions we spent and all the trees we planted, almost all of them are dead. Because all those trees everybody planted, they didn't survive. And if they did, they were just the invasive ones that we introduced. Because all those native plantings didn't work, because we really didn't understand how trees grow. We thought you just put it in the ground and it's done. The problem is, if you've killed the soil or damaged the soil, the biome of the soil, that tree stands very small chance of survival. The first drought, the first pathogen, the first insect, whatever, can take it out. So the understanding is, if we understand how this biome works and how the trees really are fed and how they interface with the fungus, when you plant a tree, you just won't plant a tree. You'll introduce the fungus and bacteria with the tree to make sure it grows. And if we're going to regrow forests that are resilient and healthy and don't get attacked by every insect and every pathogenic bacteria or fungus that gets introduced to it that wipes it out. We've got to be able to come up with a system to regrow forests so that they're resilient the way they used to be. And if we don't understand what we destroyed or damaged in the ecosystem and remodel it, we won't know how to actually do reforestation until that point. It's going to be successful. I know that's a real bummer to end on.
Yeah. No, I think that's
No,
fine.
I think it's the opposite of that, though. It's at least we're learning.
We are. The science is finally catching up to it, and we're realizing why things didn't work the way we thought they were. These public narratives that we all embrace and we all believe, turns out they didn't work the way we thought. And now the science is starting to unravel the why.
Yeah, and that's a good thing. More knowledge will come from that. I believe the paper companies have known for some time with their pulp farms that their pine seedlings, they do have to introduce some sort of bacteria and mycorrhiza to help those pine seedlings get a better.
Weyerhaeuser was way ahead of the game in doing, like I said, Douglas fir and southern yellow pine. They figured that out a long time ago, and they did it because they found out that the trees grew faster. They needed fewer years for harvest. It was an economic driver that got them to do that research and figure it out.
Mm-hmm.
But the problem is that biome is very different than the biome for oak and hickory trees up here in North Jersey.
Right.
It is interesting how the economic driver is kind of superior sometimes to getting this research done.
It always is. Economics,
Yeah.
I hate to say it, drives everything. I'm getting a little jaded about that in my old age, but it's a reality of human society that we just need to get on board with and figure out how to use it. So if we can grow mushrooms in a way that simulates how we would pick them every day out in the woods and make them available to more people and get them eating again, and their medical bills go down and their health improves, there's economics behind that. Just got to be able to get to it.
Yeah, and real quick, one thing that I didn't see here that I remember from the NOFA and J event, didn't you do a study of commercially available like mushroom pills?
No,
something
I did talk about that study. It was not us. So the top 11 selling products for lion's mane on the internet, pills and powders, were all tested. And it was really depressing. Of the 11, most of them had no lion's mane in them. And a couple of them had no fruiting bodies. They only had hyphae. They only had mycelium in them. There were only 2 of the 11 actually were made from fruiting bodies of lion's mane. So the top 11 are selling. So you can imagine all the rest of them in the market.
And I bring that up, just kind of touching on, the way that FDA regulates mushrooms, which you talked about, and
almost no regulation to date.
Well, there's no regulation for those pills and these supplements and things like that. Yeah.
Nope.
Yeah. So buyer beware.
Yeah, and there's almost no reporting on the nutritional value of any mushroom product. So I hope they'll change that. Right now, they're trying to remove the non-organic growing mechanisms to create what they're calling an organic, mushroom category. So you're not growing it on, like industrial, chemical products, and you're actually growing it on wood, and you're not spraying it with pesticides. So they are creating an organic category, but it's still not the same product as you would get in the wild or you would wildcraft.
Mm-hmm.
So actually, here's a study going on with one of the graduate students, Nick, that we know. We gave him some samples of some lion's mane grown wild, and he's comparing it to those grown in the store that you buy at Whole Foods. And I don't know if you've noticed this, but the lion's mane in the grocery store Doesn't taste like the lion's mane we grow out here in the logs. It just doesn't. But also, it looks different. It looks very different. Lion's mane grown in the wild has long teeth or hairs hanging off of it, right? The ones you buy in the store are barely fuzzballs. They got little tiny, hairs on the lion's mane. They're really short, maybe an eighth of an inch, maybe a quarter if you're lucky, but not much. But the ones that are grown in the wild, maybe an inch, inch and a half long. It's a very different look to it. It turns out, remember how I said those endophytes, the bacteria and fungus that live in the ends of trees that come out in the buds that stimulate growth? Turns out lion's mane has bacteria that live in the ends of those teeth that stimulate the growth. They're also creating more ericinase. So there's a lot of excitement around ericinase, right? That they help with regulate the growth of human nerve tissue and brain cell tissue, right? And you can, by eating a lot of ericinase, you can stimulate it. There are 16 ericinase we've discovered in wild bear's tooth and in wild lion's mane. The ones we buy in the store only have 2 or 3 of these ericinase in them. And companies are trying to introduce chemicals into the substrate to encourage these mushrooms, and this is going on in China right now, to encourage these mushrooms to generate a wider range of erinacines. I'm thinking, why are you adding chemicals to the mushroom to grow erinacines? Why don't you just add the bacteria that make it grow the erinacines? Right? But no, because they can't create the shelf life. So they're trying to chemically alter the substrates to encourage more erinacines, which you can tell if it's creating them or not by the length of the teeth on the if it's doing it naturally by the length of the teeth on the lion's mane, because the bacteria is in the ends of those teeth and it's stimulating growth there, and it's creating and the mushroom is creating a wide range of erinacines. Probably many of those erinacines are to help the bacteria do what they're doing, to make it more efficient, to clean up, to sop up ions, whatever purpose it has for creating those erinacines, we're not sure. But we know that there's more erinacines in a wider array in wild lion's mane and bear's tooth than there is in sterile grown ones. And we know that the actual look of the mushroom is different because growth stimulation is happening at the ends of all those teeth. That it stops growing very quickly in the store-bought ones. So the body of it, where the hyphae bundles are, continues to grow, but the teeth on the end stop growing very quickly if it's grown in a sterile environment. So there's a graduate student who's just started a research project around understanding that better.
Well, there's a whole lot more to be revealed here as these studies go on. So
There is.
This is interesting to get us introduced to it, so to speak.
Yep. So,
right,
so somebody just wrote, I can't get my my hericium logs to fruit. Should they be directly on the ground? I will tell you, anytime I stack hericium logs up high, they don't fruit. They like the they like the microclimate down near the ground. Most of what you'll see in the wild, when you see hericium, it's usually pretty low on a log, or it's in an area that has a lot of nearby vegetation. So it's creating sort of a microclimate. Apparently that has something to do with its fruiting.
All right.
By the way, I don't know that that's what it is. That's our hypothesis at the moment, because we've seen the same thing you have.
All right. Well, great. I'm going to shut off the recording here. I think we've gotten through all the questions. I'll stop
Great.
that.